Welcome back to the Genetics Video 2, Results of Mendel's Experiment. Keep in mind that you can always go back to the vocabulary section in case any of these words don't immediately come to your mind. So here is the very basics on Mendel's findings. Firstly, he knows that each parent is going to have two alleles for a given trait. He always crosses purebred parents in that P generation. What does that mean? It means he has one parent that is homozygous dominant, one allele capital T, one allele capital T, and one parent who is homozygous recessive, one lowercase t, one lowercase t. Now, obviously, that means that each of these parents can only give a single type of trait. So parent A can only give a capital T, and parent B can only give a lowercase t. You can see that over here on the top line. Now let's take a look at their offspring, or the F1 generation. In the F1 generation, we notice that each of them is what we're going to call heterozygous. In other words, it has one tall t from this parent, one little t from this parent, and all of the F1 generation are all going to be heterozygous, capital T, little t, for whatever particular trait we're looking at. For this one, we happen to be looking at tall versus short. It really wouldn't matter which of the traits that were presented in the last video were selected, be that P color or pod color, any of those sorts of things. It would still work out the same way, assuming that you start with a homozygous dominant and a homozygous recessive parent. So biological inheritance is determined by alleles being passed from one generation to the next. So these alleles are found on genes, and every gene has at least two alleles. For right now, we're going to only focus on two, but later on, you're going to find out that there sometimes can be more than two alleles for a given trait. But for right now, because we are talking about very simple traits and Ps have very simple traits, we're just going to talk about there being two alleles. Mendel's conclusions. The principle of dominance. What he realizes is that some traits show up more often than others. In other words, things like being tall. And think about this logically. If you were a plant and you needed to fight for sunshine, being tall is probably almost always going to be a better trait to have than being short, where you might get covered over by competing plants. So when Mendel looks at his pea plants, he knows that some of them are tall all the time, literally 100% of the time, and some of them are short all the time, literally 100% of the time. And then some of them are going to have mixed offspring, both tall and short. This is how he comes to the understanding of there being purebred, capital T, purebred, lowercase t, and then heterozygous, or not purebred, which should be capital T, lowercase t. So in other words, when a trait is dominant, it will be represented by a capital letter, and that trait will always be shown, assuming that we're talking about simple dominance, which right now we are. If it is a little t, it means that it is a recessive trait, and it means that that trait will not show up if the second allele is a capital T. Some of what I just said is now pictured here. Here is your dominant. Here is your recessive allele. Here is an example of a heterozygous or non-purebred individual. So when Mendel crosses for the F1 generation, all of the plants are tall. And all of the plants are tall because every single one of them has to inherit at least one tall gene from one parent. Because remember, he crosses a homozygous dominant with a homozygous recessive, thereby making the entire F1 generation heterozygous, but presenting, in other words, phenotypically tall. Mendel also came up with some other laws of genetics, not just the law of dominance. The first one here is the law of segregation. This should make sense to you. Think back to meiosis. When you ended up with the sperm and the egg, you know that the sperm and the egg were not necessarily genetically identical to one another. So when that sperm swims up to meet the egg. The exact sperm that meets the exact egg, we're not sure which one it's going to end up being. And so when the actual meiosis occurs and we end up with those 4 haploid cells, one of the alleles is going to be in sperm A, and one of them is going to be in sperm B. Same thing with the egg. One allele is going to be found in egg A, and one of the alleles is going to be found in egg B. So if you have a heterozygous parent, as both of these parents are, that child could be receiving capital A or lowercase a from the sperm. The child could be receiving capital letter A or lowercase letter a from the egg. So in other words, the offspring can be quite varied. And which of these actual offspring these parents bear, we're not sure, because the alleles are randomly associated with the sperm and the egg. So they could have a homozygous dominant child, 2 heterozygous children, or a homozygous recessive child. And this is going to happen for every single trait that a child receives. Obviously, children are a lot more complicated than pea plants, so most things in humans are not simply assigned by 2 alleles. But for simplicity's sake, let's pretend that we are looking at a trait that is done this way, such as things like widow's peak, rather than something that's more complex like skin tone, eye color, or hair color. Then we have the law of independent assortment. And what this means is that if you inherit trait A, or if you have the gene for trait A, it does not necessarily mean that you will receive any other trait. Having a homozygous dominant trait for pea color does not necessarily mean that you will have a homozygous dominant trait for pod color. So in other words, you can have a plant that bears a homozygous dominant pod with a homozygous recessive pea, or the other way around. So every time the organism receives a gene with 2 alleles, those are all independently assorted for. Probability and Punnett squares. I'm going to put up a YouTube video on how you do this. And so here is a very brief explanation of probability, as well as a very simple Punnett square. This is probably a little bit confusing to some of you, so don't worry. Up on Google Classroom, there's going to be a really easy to understand and really easy to follow video on basic Punnett squares. The principles of probability. The principles of probability basically say that when you have your alleles represented at the top and when you have your alleles represented at the side, each of those traits is going to go down and across and be represented in combination with all of the other traits. Heterozygous versus homozygous. Heterozygous means different, homozygous means the same. These are already words that we've gone over, and remember, you can always go back and take a look at that first slide with all of the definitions. I won't go through all of these with you. I'll just let you see that the slides are here because, again, all of those definitions we've already discussed. Genotype versus phenotype. This one is really important. Please make sure that you keep this in mind. Genotype, what are the alleles? Phenotype, what does the organism look like? This is the end of video 2.